Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Part 3, Rare Disease Clinical Development, Strategies for Ensuring Endpoint Integrity. My name is Mira, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. The presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Medpace, who developed the content for this presentation. Medpace is a scientifically driven, global, full-service clinical contract research organization providing phase 1 to 4 clinical development services in the biotechnology, pharmaceutical, and medical device industries. Medpace's mission is to accelerate the global development of safe and effective medical therapeutics through its high science and discipline operating approach that leverages local regulatory and deep therapeutic expertise across all major therapeutic areas. Now, I'd like to introduce our speakers for today's event. Meredith James is a neuromuscular physiotherapist by training and a senior member of the ATOM International team. Working as part of a team of international trainers in the development, implementation, and quality control of standardized endpoint study assessments, she trains clinical evaluators across the globe, leads investigator meeting training, and works with pharmaceutical companies on protocol development and endpoint selection and analysis. Next, Chad Leslie is a senior data manager with over 12 years of industry experience in data collection for drug development. Mr. Leslie specializes in EDC builds endpoint data management of phase 1 to 3 oncology and rare disease studies. These studies include investigational product for Duchenne muscular dystrophy, Charcot-Marie tooth disease, FSHD, immunotherapy, targeted agents, viral vector, novel biologic agents, and radioactive agents. Lastly, we've got Michelle Peterson, who has 13 years of experience in preclinical and clinical research, which includes managing global studies in multiple phases of clinical development. She has managed multiple global rare or ultra-rare disease trials with a focus on neuromuscular and pediatric disorders. Ms. Peterson is a member of Medpace's Specialized Rare Disease Consortium, and she is adept in developing strategies necessary to address the potential challenges of recruitment and logistical requirements for studies of this nature. And now, without further ado, I'd like to hand the mic over to our speakers. They may begin when they are ready.
Thank you, everyone, for taking the time to join our talk about endpoint integrity today. As we all know, rare disease trials can be particularly challenging when it comes to endpoints. Not only are we typically limited in the number of patients that are reasonable to include in the trial, but often the endpoints themselves are novel or complex. Today, we'll be talking about years of collaborative research that has optimized training and oversight of pulmonary and physical function testing, which can be applied to other complex endpoints. The collaboration between subject matter experts such as ATOM, biometrics, medical, and operational teams has resulted in better training, faster startup, and data visualization that has made it easier to detect patterns and issues. So without further ado, I'll hand it over to my esteemed colleague Meredith James to tell you more about ATOM and how they've worked to protect endpoints.
Thank you, Michelle. I know Dr. Michelle Eagle was billed as the speaker today, but unfortunately there's been a bereavement in her family, so I'm standing in as a senior member of the ATOM team to give this presentation. Next slide, please. So in the talk today, I'm going to take you through who ATOM is and how we came to be, what it is that we do within clinical trials, outcome measures and endpoint, our processes of quality control, and the results. Next slide, please. ADAM International was founded by Dr. Michelle Eagle, who's a PhD physical therapist in the north of England over 10 years ago. Michelle's been working on clinical trials since the first trials started in neuromuscular disorders and has over 30 years experience as a physiotherapist. Our team comprises of 18 physical therapists from around the globe, and as you can see from our map with the green flags, we're based in Australia, Europe, North and South America. Several of our team members are global key opinion leaders in neuromuscular disorders and clinical endpoints, clinical research. Next slide, please. Our indications. We work predominantly across neuromuscular disorders, including the main conditions within this rare disease group, so Duchenne, Becker, spinal muscular atrophy, limb-girdle muscular dystrophy, FSHD, and myotonic dystrophy. We also work across glycogen storage disorders such as Pompe disease and Farber disease. We're involved in trials with mitochondrial disease, which is an emerging treatment group. We do a lot of work across achondroplasia and skeletal dysplasia studies. More recently, our work has expanded into studies that are based in the intensive care or intensive treatment unit, and we also work in the autism area.
Absolutely. And Medpace and ADAM really have been able to enhance training and oversight across these indications by employing successful strategies that we'll detail later on in our talk. But we found that lessons learned in one disorder can easily be transferred into another.
Next slide, please, Michelle. The first clinical trials in neuromuscular disease commenced around 2007, and there was just one trial in that time, and now the field has exponentially expanded to the point where we have about over 40 trials in this area now. And at that time, the disease-specific endpoints or outcome measures were not available. We were just running with what we had. And at that time, for the first trial, there wasn't actually any clinical evaluator training to standardise endpoint data collection. The next study that came about in the field, we did have an investigator meeting together with all the medical and nursing staff, and then everyone went back to their sites, and it was a global study, and there was no checking up on how the data was being collected. If new staff members came to site, there was no training for them. They just picked up a manual and tried to go with it. And so we did have a lot of difficulty in achieving primary endpoints because there was so much variation in how sites were collecting the data. You know, we think when we work across the medical field that we all do things the same way, but we don't. And it's not until we all come together, and it's been a really iterative process over the last 13 years in how we have developed a package of standardising training, standardised manuals of operation of how to collect the endpoints. And then within the endpoints themselves, we needed to have a rethink because we were initially borrowing from other disease groups. For example, the 6-minute walk test was borrowed from the cardiorespiratory area and adapted into neuromuscular. And so over that time, we've now got disease-specific endpoints. We have a wealth of training materials. We're involved from the outset of a trial. So in the early stages of protocol development, we're able to advise on endpoint selection. And we work very closely with Medpace in developing our training and data collection methods. And so ADAM is now the industry leader in advancing trial outcome measures. Next slide, please. So ADAM's involved, like I said, from the outset. So we're involved in providing consultation on protocol development, including the target population, inclusion and exclusion criteria in order to try and homogenise the population, looking to endpoint information and guidance on endpoint selection, development of a clinical manual of operations, so to standardise the collection of data throughout sites. And most of our studies are global, multi-centre sites, so this is hugely important. We train all clinical evaluators who will be collecting those primary and secondary endpoints. We develop, in conjunction with Medpace, source documentation that collects the data, but also minimises any chance of error from the clinical evaluators completing it. A huge part of our work is data quality control via video reviews of assessments. We also have a good camaraderie with our clinical evaluators. Each clinical evaluator for each trial has a named ATOM member who is their personal buddy that they can email or phone to give them support throughout the course of the clinical trial. And that may just be clarification on the testing procedures. It may be they've had a difficult patient and they need some advice on how things can go better. And that ATOM member is there for them throughout the course of the trial and always available. We also are hugely involved in data review, and Chad will take us through that process a little bit later on. Next slide, please. So why do we need all these mechanisms of quality control? Well, as you're aware, our evaluation of the endpoints or outcome measures, I use the terms interchangeably, in trials are a serious and expensive business. The patient groups we are looking after are living in hope that we may use these clinical trials to provide robust evidence and potentially treatments for them. A good outcome measure or a good endpoint is reliable, it's repeatable, and it's standardised. And that same endpoint also needs to be sensitive enough to measure change over the period that a trial's set. In order to ensure that if you pick the right endpoint, in order to trust the data, you need to have data collection which is dependent on high quality, reliable, standardised evaluations that are collected by a trained physiotherapist or clinical evaluator. So how does ATOM facilitate these factors? Next slide, please. From the outset, we have our consultancy on protocol design and endpoints. We are heavily involved in advising pharmaceutical companies on which endpoints might be an appropriate choice for their population and for the drug mechanism that they're reviewing. So we can advise on the population, the endpoints, inclusion and exclusion criteria, the schedule of events for the study, the training plan and potential IRB implications that will have, what quality control measures need to be put in place, not only for the training of the clinical evaluators, but for data and data quality throughout the study, and involvement within this ECRF and source documentation construction. Next slide, please. I generally talk about physiotherapists as clinical evaluators because when we're taking measures of functional performance, strength, respiratory function, these measures are generally performed by physiotherapists in the main. And so they gather the functional trial endpoints. And for most of the trials that Adam's been involved in, the physiotherapists have been responsible for the disease-specific endpoints that are used in current trials or standardised endpoints in order to have them better used within clinical trials. However, the level of a physiotherapist training around the globe can be quite variable, and their level of autonomy as well. And so this is why Adam has an important role in providing education and support and training to any clinical evaluator within a clinical trial so that each clinical evaluator is confident and competent and reliable and standardised in their data collection. Next slide, please. Our training is based around delivering standardised and reliable and repeatable clinical evaluation. That way we minimise the variability between study visits. And so if we do see change in the data, we know that it's a true change and not variation in the performance of the clinical evaluator or potentially the performance of the participant. They're responsible, the clinical evaluators, for the collection of the data based upon a standard manual of operations, and they must follow that manual to the letter to reduce any internal validity risks that are associated with effort-dependent tests. Generally, any of these endpoints, which could be the spirometry or a walking test or time to get off the floor, these are effort-dependent tests. And so they require motivation of the clinical evaluator and the participant to get the best performance on that day every time. Next slide, please. How do we deliver training? Well, here's Michelle delivering some training specifically around an item of a scale that uses a step, and how standardization is hugely important. So we deliver our training in a number of methods, but it's always individualized to the needs of that person or group that we are training. We can attend at investigator meetings. We have also worked with Medpace, providing physiotherapist-only meetings. The team, we do site visits. We can do WebEx. We do the buddy support, as I explained earlier. So every named clinical evaluator will have an ATOM member that they are able to contact. And in the event that they can't get in contact with their first named buddy, they have a list of the other team members to contact. We also ensure that we have the manual of operations in a language that is appropriate to the clinical evaluator's native language. And we also work with Medpace in developing source documentation that facilitates a smooth and accurate assessment. Next slide, please. So which outcome measures do we train? Well, here we have a picture of a young man who's doing some spirometry or some respiratory testing. So we do a lot of this. The traditional means of a forced vital capacity or a peak expiratory flow using a number of different systems. We look at more modern measures such as mouth inspiratory and expiratory pressures. And we also have the sniff test, which is a sniff nasal pressure, which can relate to diaphragmatic function. We teach across disease-specific and general functional motor performance scales. So that may include for Duchenne, the North Star Ambulatory Assessment, which the members of our group developed, or the Performance of Upper Limb, which we also developed. So the ATOM training team, physiotherapists that not only do training, but are also clinicians, expert clinicians working in clinical trials as clinical evaluators as well. We teach on time tests, we teach a number of methods of strength testing, we teach upper limb motor scales and upper limb scales of dexterity, such as a 9-hole peg test. In our anthropometric measures, we can teach measurements of height, ulnar length, other segment lengths, arm span, circumferences for skeletal dysplasia studies. And what we're very aware of is that while some of our clinical evaluators have excellent clinical experience, that doesn't necessarily make you a perfect clinical evaluator within the trial environment. And so that's why we developed these standardized training procedures and manuals of operation to ensure that we minimize variation in the data and that data change is true change. Next slide, please. ATOM works with Medpace very closely in working on our documentation, looking at how we make the source documentation best reflected within the ECRF, our training materials, so for delivering training, and also the manual of operation. We ensure that we have certificates of training that are delivered to site, and then also back into central files. We have video quality control of assessments, which I'm going to talk about in a little bit more detail. And with Medpace, which Chad will talk about, we have data review meetings where we're looking quarterly at the data to see what trends and patterns we're looking at. Next slide, please.
Yeah, thanks for the shout out, Meredith. I just want to just reiterate how crucial it is to have this standardized CRF library for these disease-specific endpoints. It makes it easier for the sites to enter the data, and then makes it easier for us to present the data during our data review meetings. Thanks.
Absolutely. So the process of where we're at now, sort of 10 years later, it's been an iterative process. And one of the biggest things we've learned is how important video quality checks are during the course of the clinical trial. So what this involves is that a clinical evaluator at predetermined time points will take a video of their assessment, and that allows us to then comment on that and reduce variability of the assessments by providing immediate and timely feedback to that clinical evaluator and that site on the performance of their test. We can look at how the assessment was delivered and ensure that it's meeting the standard. It lets us know that the assessment's been performed correctly. We can change the positions that the assessors are doing the tests in. It's a really nice way to keep everybody feeling part of the global team of a study when they're getting positive reinforcement that they're doing a great job and they're looking after their subjects really well. And it's a continual training, so we don't just leave our clinical evaluators after a investigator meeting to go and do their assessments and be subject to data drift, which we know previously has been a problem. And so this video quality control has been essential actually to looking at data quality throughout the study. It does let us capture invalid assessments and data, and why this is really important is that when we look at scheduling visits for a protocol, having a screen visit prior to baseline and randomization and having that screen videoed is essential. It allows us to see any errors specifically related to inclusion or exclusion criteria and have those corrected before the baseline visit, ensuring that you have the most appropriate population entering the study according to the guidelines. And the mechanism of doing this is we use a company called Chili Farm, who are present in over 40 countries and have got through the regulatory requirements and privacy laws to allow a clinical evaluator at their site to upload the video of their assessment to a secure portal where the ATOM team are able to review that and in conjunction with the Medpace team provide the feedback, file the quality control reports and make changes as required.
Absolutely. And together with ATOM's help and Chili Farm, we've been able to enhance our competent authority applications with robust privacy information as well as rationale for why the videotaping should take place in more than 40 countries. And to date, we haven't had any issues with that ultimately not being permitted, but we've found that this really streamlines the application process to take these concerns that we've heard from the authorities and make sure that we address them in the initial application.
Okay, so with our experience, thanks Michelle, with our experience of QC, what do we do about it? And this is a team approach between ATOM and Medpace. The first thing the video assessment allows us to do is to actually see who's doing the assessments, and then that makes sure that they are someone that has been certified by ATOM as being trained for that specific trial. It also lets us see wrong starting positions, and where this can be really important is if you use something like climbing up 4 stairs as an inclusion criteria, and you have a set time that study participant needs to perform that test, and the clinical evaluator has used the wrong starting position, then the test time is not going to be accurate and it could compromise their inclusion into the study. We've also seen unsuitable testing areas. So particularly for something like the 6-minute walk test, which requires a 30-metre corridor, despite site initiation visits and site selection visits, sometimes things can go a little rogue and or things can change at site, and we've had unusual areas that these tests have been performed, including the aisle of a church and underground hospital car parks, top of the buildings, and outside. And so it is, in order to maintain data quality and standardization of the testing throughout the study, we need to know what these environments are and to ensure that they are the correct environment at every visit. The videos also allow us to look at the order of evaluations to ensure that they've been carrying out correctly. A lot of these studies involve multiple assessments of children and adults, which are fatiguing. And so if you don't do the assessment in the correct order, you introduce a fatigue bias into your data. And so that's a really useful tool from video as well. Next slide, please. I've borrowed my son just to show some of the errors. We are trying to do standardized assessments and standard data quality standardized data quality throughout the globe in multiple sites. And so something as simple as the study equipment can have a huge impact on the data and the assessment performance. And Michelle's going to talk through that shortly. But just to illustrate, even this test as simple as standing up from a chair, if you use a garden chair with a sloped bottom and armrest, that's going to be very different to using a standardized hospital bed or plinth or bench, because you can't adjust the height. And in this case, we perform that test without armrests, and there's armrest presence on that chair, which will change the test for that child. In the middle picture, unfortunately, the chair has wheels, and this is something we have seen before, where a sitting to standing test has been performed at the correct height, but the chair has wheels, and that is a massive safety concern for a child or adult who has profound muscle weakness. In the last picture, we're looking at a child's initial position before they perform a rise from floor, which is a very common assessment in our field. And the child's wearing socks, he's on a cushioned mat, and he's not in the correct starting position. And so we have a safety concern from the socks being on. We have a floor surface that's inappropriate. And by not having the correct starting position, that's going to impact on the time that that boy performs that test. And when we're looking at small changes in time over the course of a year, having an error like this could impact the time by 0.2 or 0.3 of a second, which is going to compromise the data. And so these are just little things that we can control to help make a trial's data the best quality that we can. Next slide, please. And so with Medpace, we work together when we do the QC report to look at whether we classify a deviation as a minor error that's not going to affect the scoring, but the test could be done better, potentially retraining if it's a persistent minor error, or major errors. So like I described earlier, where we have potentially something like a 4-stair climb being performed incorrectly, which is an inclusion criteria. And so there's a number of things that can happen as a result of seeing those errors. Next slide, please. ADAM is a global team, as you saw earlier, from where we're based. These are the countries we work across. We work across over 40 countries. We speak English and French, Italian, German, Dutch, Spanish and Portuguese, and a little bit of Japanese. So we're well versed in a number of languages, and we have a very good community of clinical evaluators around the globe. Many of our evaluators work on multiple trials, and so we have a good camaraderie and an end focus of taking the best evaluations we can on our subjects' behalf to keep a trial full of robust and reliable and repeatable data. And I think that's my last slide.
Hey everybody, this is Chad Leslie with Medpace Data Management. So about 6 years ago, we started working with the ADAM group on a couple of clinical trials, and what we started doing was reviewing the data periodically to see if there were any signal in the data that could indicate an issue with how the tests were being performed by the clinical evaluator at the site. So what we did is we set up quarterly data review meetings, and for those we provided subject profiles with listings, including any kind of data that we thought may affect the endpoints, may affect the physical function tests that were being performed, including demographics, the tests themselves, any adverse events or concomitant medications the subject might have been on, and then any other procedures that may have confounded the clinical evaluator tests. We also provided data visualizations to the team, and these can be done in Spotfire or even Excel or Tableau, and we'll show you a couple of those here in a minute. It is really important that when we set up these data review meetings, that we pull the data, we extract it for the visualizations and the patient profiles, that the data be as clean as possible. Hopefully that's source data verified, but a lot of times we're pulling data before we can get a CRA out to the site. And that's okay because we've got electronic checks built into the system that are going to fire to kind of work through as much of that dirty data as possible. but sometimes you just don't have the luxury of having clean data. So we like to get it pulled and get it looked at as soon as possible to catch any kind of problems at the site. We like to get out and, well, we like to have our first assessment after the subjects have had 1 or 2 visits. So you know, once again, that we can find any errors as quickly as possible. So we're going to kind of walk you through what we see at one of our data review meetings. So our first subject here, if you could see the top left here, we've got our 6-minute walk test graph. So at the x-axis, we've got our over time, those are our visits occurring, and the y-axis is the distance the subject walked in 6 minutes. So as you can see, this subject is kind of, the distance is declining over time. It's a pretty standard, you know, decline there that we would kind of expect to see in this indication. So hopefully the subject's in your control group. If not, you might have problems. Sorry, a little data joke there. So the other tests we have are the 10-minute walk test below that. For that test, the y-axis is actually in seconds, and we've inverted the axis. So actually, it takes, let's see, the performance is actually declining over time. It's taking longer for them to do the test over the course of the event. So we reversed the axis on the time test so that they would correlate with the 6-minute walk test. So the 6-minute walk test graph is declining. We would also expect the time function test to decline as well, and that's true for the Stanford supine for the top right, and the 4-stair climb, and the 4-stair descent. So this is typically what we would see in an average subject. I don't think there's any indication of any clinical evaluator error here. Let's go on to the next slide. So for this subject, we're starting to see a little problem here. We've got our 6-minute walk test. The subject is improving over the first few visits there. Then we come to visit 3. At visit 3, we're dropping about 58 meters. And then at visit 4, it pops back up to about 83 meters. So we can clearly see that visit 3 measurement, there's something off there. It doesn't fit in with the other expected measurements for the test. So if we look at our 10-meter walk run, the subject is improving over time. And then, so there's no indication of anything there. There's Stanford supine. There are other function tests. There's a little bit of variation there, but we have a very small window there. We're only talking about, you know, half a second overall variation. So I don't think there's much of a indication of anything there. All right. So we definitely have an indication there at visit 3. So what we did then, we kind of broke it up with the actions that we would take. The clinical evaluator team would go back and look at that visit 3 for that subject. Well, they'll look at the video to see if there's any kind of issue. Maybe they changed the location of the test, or, you know, clinical evaluator maybe changed at that visit. And then the data team's going to go back to the site and ensure that data point wasn't just entered incorrectly. Maybe we had a transposition of the numbers when they did the entry, but we'll go back and follow up with the CRD and make sure that data is actually correct. Another thing we can do is to review all the rest of the subjects that are at the site. So we'll go to the next slide. Okay, so for this slide, we pulled in all the rest of the subjects at the site. And if you look at the 6-minute walk test, you can see most of those subjects are having a decline at visit 3. There's definitely an indication that something's going on there. The rest of the test seem to be fairly flat and even. So I think the only signal we're seeing here is that 6-minute walk test. So I think then we kind of go back and ask ourselves, was the test order switched? If they performed all the other tests prior to the walk test, we definitely see, you know, maybe some fatigue in those subjects. So that would show up video, and in the CRAs, we'd also be able to follow up with source documents to find out what order those tests were actually done in. We can also find out if course location was changed, were the subjects wearing shoes or were they not wearing shoes? That could affect the test as well. And if a different clinical evaluator performed the test, we've definitely seen where new evaluators come in and results have been changed just based on who is doing the test.
So those certainly, Chad, have been lessons we've learnt over the last 10 years and put processes in place to try and minimise examples like this by standardising everyone's training, teaching them how to use the source documentation correctly, putting in checklists within the source documentation to try and prevent any of these errors and keep the tests as standardised as possible.
Absolutely, and these same visualization strategies have also been utilized in other endpoints, such as imaging lab oversight. So this has been a really helpful tool just to make sure that we are spotting outliers and addressing these challenges as they arise.
Great. Thanks, Michelle, Meredith. And that's all I have for today. I'll turn it back over to you, Michelle.
Thanks, Chad. All right. So I'll be talking a little bit about operationalizing these items. One of the most important hallmarks of success with executing complex endpoints is to build systems with your plan in mind. As Meredith mentioned earlier, endpoint integrity is enhanced when patients are completing procedures in a certain order, so that impacts from fatigue and other items are accounted for. By simply arranging worksheets and CRFs in the recommended order of procedures, compliance becomes more intuitive and data entry swifter. Early protocol collaboration helps ensure the best endpoints are selected and reasonable to operationalize. A common issue we spot is too many assessments to be reasonably completed during business hours. By strategically placing important endpoints earlier in the day, we can help enhance capturing the patient's abilities accurately. We also have found that optimized communication can help accelerate study startup and ensure all parties are thinking about oversight on a consistent basis, not just at the beginning of the trial or right before a database lock. Regular meetings to ensure alignment of contracting and approvals with study initiation, visit, and endpoint training have been important to ensure these are not rate-limiting items. And on the next couple of slides, we'll discuss training and study equipment. Not only do the ADAM team have a wealth of experience, as you can see from these 200 years of industry experience, they are also involved in developing the endpoints themselves. They've made us an optimal partner with Medpace. Medpace has developed a rare disease certificate program from a cross-functional team so that all team members working on rare disease studies benefit from our institutional knowledge and innovations. This program includes a monitoring exercise for our CRAs so they can be exposed to common challenges, such as complex medical history or home health care issues. This extensive training builds a great base for the additional study training they receive upon being assigned to a trial. Our CRAs are also highly trained to build positive relationships with sites to encourage open, honest, and timely communications. They understand the amount of time and effort it takes to onboard a new study team member, so they work to give the operations and ADAM teams as much advance notice as possible when there are changes to site staff. Their understanding of the importance of study equipment have also detected and prevented countless issues. While providing sites with study equipment seems like a simple task, there are many ways to ensure it doesn't become an issue for your study. For example, it's important to ensure equipment is approved for use in the country you'd like to conduct the trial. Having this knowledge in advance can help you pick appropriate equipment and develop an appropriate regulatory strategy. We've also learned in our early trials that while it's wonderful we can procure a 4-step staircase built to study specifications, we need to make sure it can fit through the door upon delivery. That actually happened. Importation planning will also make sure that study equipment arrives on time and doesn't become rate limiting for startup. Additionally, planning on what you'll be doing with study equipment at the end of the study is important to address at the very beginning of the study to avoid issues like shipping charges that are several times higher than the value of the provided item. And we like to address those items in the contract upfront so that there's a plan. After doing countless investigator meetings together with Adam, we've been able to deliver quality hands-on training in an efficient manner because we've already done things like develop specifications that allow for a 6-minute walk test to take place on a 30-meter track in a hotel in Europe or wherever else in the globe. With rare disease, we often have to have a global reach to obtain the required sample size, and we want to ensure the site staff performing the procedures understand them to the letter. So as Meredith mentioned earlier, we've worked to finalize manuals very early in the process to allow adequate time for translations and ensure that we can procure translators for investigator meetings or on-site trainings. And I'll let Meredith explain our lovely photo here.
So I think this is just a timely reminder of ensuring when you're starting up with particularly a trial-naive site or country, the expectation of what equipment might be arriving for the study, and ensuring that you have an appropriate room in which evaluations can be carried out. We find obtaining photographs from sites before Medpace or Adam staff can be on site to be very useful to ensure that we do have the correct space in which to obtain the primary endpoints for studies.
Thanks, Meredith. So in conclusion, a lot of strategies we've put together seem logical and effortless, and you may not notice a difference if you haven't worked in the old world 10 years ago, where we were pioneering some of these techniques. The difference really is less issues arising due to this proactive approach. When issues do arise, we're able to detect and correct them earlier to protect the trial's endpoints. We'll continue working hard on this continuous improvement cycle to deliver the best trials possible for our patients and partners in research. Thank you.
Thank you very much for that insightful presentation. Now, I would like to invite our audience to continue sending their questions or comments right now using the questions window for the Q&A portion of this webinar. I've already received some questions, so I'll start with those. Our first question here is, the question's for Meredith. How do you manage training multiple nationalities at IMs?
Oh, it's a lot of fun, to be honest. By being very well organised, by working with Medpace ahead of time to understand which countries and which languages are going to be spoken, by bringing in simultaneous translators, so booth translation, by grouping, our training is very practical training, teaching people how to do an assessment. So that can supersede any language barriers. But we bring the Adam team obviously speak a number of languages, and we tend to group our common languages together into small groups when we're doing small group training. So within an investigator meeting, when we have a breakout session to teach endpoints, even within that breakout session, we'll go into small groups with an Atom trainer with that group to try and cover all the languages. And if we can't cover the language ourselves, then we have the simultaneous translator present. And because it's very practical application, that assists us too.
Thank you very much for that answer. Our next question is it possible to have an edit check identify clinical evaluators not performing the tests correctly?
So in terms of looking at how, sorry, was that for me?
Sure, go ahead, Meredith.
For us, it's a combination of things, but the video control, the video quality control is certainly the main visual method that we have to see in conjunction with the documentation that's provided by that site.
Hi, and this is Chad. I'll just jump in here. We do have edit checks built in the system to look at variability across, well, we don't, not really across visits. So that's the issue. We can look at maybe a range for a test to make sure that a value is within range and make sure that it makes sense for that test. So we have edit checks that are simple on the visit basis, but we can't really look across all the visits like we can when we're looking at the graph. So it's important to have that data review piece there as well, and have the clinical evaluator group, have the Adam group actually looking at the data, because they've taught us quite a bit about what it means when we're seeing the data spike and jump around like it does.
Thank you very much for that answer. Our next question is, what has been a common challenge in operationalizing physical function endpoints?
I'll take this one. This is Michelle. So I think commonly we see in naive sites potentially not having the appropriate person for folks to actually take on the tasks in the clinical research trial. So if we are doing a lot of assessments that are best done by a physical therapist, we want to make sure that they do have the education of a physical therapist and the right background to actually start on the trial. And we can often help sites with identifying people if they don't have someone on site that fits that build. But just starting with the right people is the best first step. And then after that, our training and oversight plans, as we've detailed earlier, is really key.
Thank you very much for that answer. Our next question is for Meredith. What other types of training do you offer other than clinical trials?
So Adam works across natural history studies as well. So even if we're not looking at investigational product, we train clinical evaluators globally for natural history studies. We've also been involved in standards of care workshops, looking at sort of emerging countries that aren't yet involved heavily in clinical trials, but maybe in the future, and looking to upskill within that country the knowledge around a condition, common assessments and endpoints.
Thank you very much. Our next question here is, for video QCs, is it only at CE administration, or is it for scoring of the items too? If it is for scoring, how do you go about changing the data or overall scores?
So the video QC is just as an overview of the administration of the assessment. For instance, if we're looking at the North Star ambulatory assessment for Duchenne, we are not changing the clinical evaluators' scores. We're looking at just at their administration of that assessment and ensuring they're using the correct positions.
Thank you very much. Our next question is, do you have experience conducting assessments via video in patient homes?
Well, in the current age of world global shutdown and COVID, this is a very acute area at the moment that we are working very rapidly in to look at how we can protect trials that are already underway in collecting that data. And so we've just recently, only in the last 3 or 4 weeks, been working very hard to develop protocols and look at which endpoints are actually suitable to be carried out within the home environment via a video link with the family, and how that data potentially can fit in as study visit data within the whole duration of the trial. So it's a very new emerging area, and I think over the next sort of 2 or 3 months we'll have a much clearer idea about our experience with that.
Thank you very much for that. Our next question here is, have you found effective ways of dealing with research teams who make the same errors related to data collection multiple times despite retraining? Rare diseases have limited patient population, so every data point counts.
That's absolutely correct. So I think this is a team approach between Medpace and Adam, depending on what the challenge is and where the error is occurring with which teams. So when we see these types of things, this is a topic of discussion that we tackle with our sponsor, Adam, and Medpace together to try and understand the root cause of these repeated errors and what might be the best strategy in order to prevent it from happening in the future. And then, of course, implementing that strategy action plan with who's going to do what action, as well as how it's going to be documented is how we handle those types of items. Meredith and her team have been really kind in physically going to the site if that's necessary, and the Medpace staff members are also available to do that. We also have physicians that work really hard to assist from a medical perspective as well, to have some of those difficult conversations in a doctor-to-doctor manner if that's where part of the problem is occurring.
And I think onwards from that, Michelle, I think we try and anticipate those issues by being very well organised at the beginning of the trial, and like you say, bringing in the correct professionals to be involved in the trial and collecting that data, and ensuring that people understand the protocol, understand the manual of operations is there for a reason, participate in a standardised training. But as Michelle said, we can return to site. I think one of the other things that's been most useful in protecting data has been the introduction of reliability testing prior to a site first patient visit, and that's whereby we bring in a patient volunteer with an ADAM staff member and the site CEs team, and we work with assessing that patient together. And so the CEs have had the opportunity to perform the assessments before the pressure of a screening visit, and so we can iron out any mistakes before the first patient visit is due.
Yeah, that's a great point. That reliability testing has been really instrumental. Thanks, Meredith.
Thank you very much for that answer. Our next question is, when test isn't performed according to MOP, do you file a deviation, note to file, or others, especially if the deviation is minor?
This is a really great question that doesn't have a very simple answer. So again, what we do is we at Medpace have a plan for protocol deviations outlined on what constitutes a protocol deviation and how it's classified and how that is recommended to be documented. We do often see the manual of procedures as an extension of the study protocol. So often we are capturing things that are being done incorrectly as deviations, so that if there are any impacts to the data, there is a corresponding explanation as well as what's been done to prevent it in the future documented. So in a lot of cases, these do end up being deviations, but if it is something that is outside of what we expected and planned in advance in our protocol deviation plan, then we would again bring this back to the ADAM, Medpace, and sponsor team for discussion on how it should be documented, retrained, and addressed with the site. Anything you want to add, Meredith, on that one?
No, I think you've covered it there, and I think this is why we're sort of developing, the checklists around what's a minor issue on video control, which won't affect the data that's recorded in the source documentation versus a major deviation, such as the wrong position on an inclusion criteria test that would, necessitate a rescreen or a repeat of that test. But good documentation from the outset with a documentation plan is essential.
Thanks, Meredith. Wow, I'm really impressed with these questions. It's a talk about data, which sometimes can be a little bit dry, so we really appreciate everyone's attention and great questions. Mira, I'll hand it over to you.
Thank you. On that note, we have now reached the end of the Q&A portion of the webinar. If we could not attend to your questions, the team at Medpace may follow up with you, or if you have any further questions, to please direct them to the email address that's showing on your screens. That's info@medpace.com. Thank you everyone for participating in today's conference. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screens. Your participation is appreciated as it will help us improve our webinars. Now, please join us in thanking our speakers, Meredith James, Chad Leslie, and Michelle Peterson, and feel free to share the recorded version of this webinar using the link in the chat box when it becomes available to you. We hope you found this conference informative, and have a great day, everyone.